You know that feeling, right? That sudden jolt before a big presentation, or the surge of energy when you narrowly miss an accident. It's the adrenaline rush. But have you ever stopped to think, what is actually going on inside your body, inside your very cells, during those heart-pounding moments? Well, today, we're going to dive deep into that molecular alarm system that's so crucial for our survival.
So what exactly is an adrenaline rush? I mean, we all know what it feels like, but what's the deal on a biological level? It's definitely not just some vague feeling. Nope. It's an incredibly precise, lightning-fast response to a signal from the outside world.
Yeah, you know the signs. Your heart just starts pounding in your chest, your palms get all sweaty, your senses feel like they've been cranked up to 11, and you get this insane burst of energy. These are the classics, right? But what's wild is that all of this, it's all just the downstream effect of one single type of molecule kicking off a massive chain reaction inside you.
So that's the big question, isn't it? How can one tiny little molecule cause all that chaos? Well, to figure that out, we have to follow the trail. We're going to trace the journey of a single adrenaline signal, right from the moment it shows up at a cell's doorstep, all the way to the action it triggers deep inside. Okay, so this diagram here, this is basically our roadmap for the whole process. And yeah, I know it might look a little intimidating at first glance, but trust me, it's not that bad. Just think of it like a molecular relay race. Seriously, each part of the chain just hands off the signal to the next, and the whole thing happens in like a fraction of a second. And this whole relay race, it actually has a name. It's called signal transduction. And really, all that means is it's the process of turning information, like adrenaline showing up, into a real chemical change inside the cell. And get this isn't just an adrenaline thing. It's a fundamental process that happens in pretty much every living cell on the planet, from tiny bacteria all the way up to us. So how does this race actually work? Let's break it down step by step. First up, you've got adrenaline. Its scientific name is epinephrine, and it binds to its own special receptor on the outside of the cell. It's like a key fitting perfectly into a lock. Once that happens, it activates a middleman called a G protein. This little guy then zips over and switches on an enzyme. That enzyme, adenylocyclase, then starts cranking out tons of an internal alarm signal called CAMP. And finally, that CAMP activates the real workhorse, a protein called PKA, which goes off and carries out the final order. Let's zoom in on that middleman for a sec, the G protein. The best way to think about it is like a molecular light switch. So when it's in the off position, it's holding onto a molecule called GDP. You can just think of GDP as a dead battery. But when the adrenaline receptor gets triggered, it flips that switch. The G protein literally drops the dead battery, GDP, and picks up a fresh, fully charged one called GTP. And that simple swap is everything. It changes the G protein's shape, turns it on, and sends it on its way to the next step in the relay. Okay, so remember that enzyme adenylocyclase? Once it gets activated, it just goes nuts. It starts producing huge amounts of this molecule CAMP. And here's a great way to think about it. If adrenaline is the first messenger, the one who arrives at the castle wall with an urgent message, then CAMP is the second messenger. It's like the town crier who runs through the streets ringing a bell and spreading that alarm to everyone inside. It's how the signal gets broadcast far and wide throughout the cell. And this, right here, is where the story gets absolutely incredible. It is all about amplification. I mean, how can just a handful of hormone molecules create such a massive, body-wide feeling? The answer? It's a domino effect, but on a scale that is just staggering. What you're looking at here is something called an enzyme cascade. And check out how this works. One single activated receptor doesn't just activate one G protein. Oh no, it can activate hundreds of them. And then each of those activates an enzyme that goes on to make thousands of CAMP molecules. So with every single step, the signal just multiplies and multiplies, turning a tiny whisper into an absolute roar. Okay, let's put a number on that roar, just to see how crazy this is. The binding of a single molecule of epinephrine, just one, can lead to the release of, wait for it, 100,000 molecules of glucose into your bloodstream. 100,000. No, that is not a typo. One molecule unleashes 100,000 units of pure energy, getting your body ready for whatever's coming. So the next time you feel that sudden, totally overwhelming rush of energy, you'll know exactly why it feels so powerful. It's the sheer mathematical power of amplification at work inside you. The whole system is perfectly designed for maximum impact from the tiniest possible signal, getting you ready to fight or flee in an instant. But, of course, that feeling, that super intense feeling, it doesn't last forever, right? Thank goodness. If it did, we'd just completely burn out. So it turns out that the off switch is just as important, just as cleverly designed as the on switch. The system has to have a way to reset itself.
I mean, it's the obvious next question, isn't it? If this whole cascade is so explosive, how on earth do you turn it off? Well, it turns out the shutdown mechanism is every bit as elegant as the startup process.
Here's the deal. After a receptor has been active for a little while, it gets a kind of chemical tag put on it. And this tag acts like a beacon, calling over another protein. And this one has the perfect name for its job, beta-arrestin. And yeah, just like the name says, it literally arrests the receptor. It latches on and physically blocks it from turning on any more G proteins. Boom, the relay race is stopped right at the source. And just to make absolutely sure the signal is dead, the cell does a little bit of housekeeping. The whole receptor-arrestin complex gets pulled inside the cell. It's a process called endocytosis. So by basically taking the receptor off the market for a little while, the cell guarantees it won't get accidentally triggered again. Then, later on, it can recycle it back to the surface, completely reset, and ready for the next alarm. All right, let's zoom out for a second, because this is where things get really mind-blowing. This whole incredibly sophisticated system, the receptor, the G protein, the second messenger, it's not just for adrenaline, not even close. You see, biology is the ultimate recycler. It reuses the same brilliant molecular toolkit for tons and tons of different jobs. The best way to think of it is like a modular design, kind of like Legos. Nature basically perfected this one signaling module and then just started plugging it in everywhere. Seriously, the same basic machinery that gives you an adrenaline rush is also responsible for your ability to see, to smell, even to taste. And this right here shows that comparison perfectly. On the left, you've got our adrenaline pathway. And on the right, that's the pathway for vision. Just look at the similarities. It's uncanny. In both, you've got a receptor, you've got a G protein, and you've got an enzyme messing with a second messenger. Sure, the names are a little different, rhodopsin here, transducin there, but the fundamental logic, the core engine is exactly the same. It's amazing. Now, because this system is so universal, so widespread, that also makes it a potential point of failure. I mean, if it's used everywhere in the body, what do you think happens when a critical part of it, like that all-important off switch, gets stuck or breaks? Well, that brings us to a really nasty disease, cholera. The toxin that the cholera bacterium makes is basically a molecular saboteur. It sneaks into your intestinal cells, and it targets that G protein. It chemically modifies it in a way that breaks the off switch, locking it permanently in the on position. And the result is a signal that just won't stop, causing the cells to pump out insane amounts of water, which leads to that severe, life-threatening dehydration. But here's the flip side. This works both ways. If these pathways can be hijacked by disease, well, that also means we can target them with medicine. Because we understand how they work, we can design drugs that intentionally flip these same molecular switches for our own benefit. A great example is a really well-known drug, sildenafil. It works by targeting this exact kind of pathway. But what it does is pretty clever. It doesn't turn the signal on. Instead, it blocks the enzyme that normally acts as the cleanup crew for the second messenger. So by stopping the cleanup, it just makes the on signal last longer than it normally would. It achieves its effect simply by extending the signal's duration. Pretty smart, huh? So think about it. From a life-saving jolt of fear to the simple ability to see a beautiful sunset, it all comes down to these incredibly intricate, silent molecular conversations. Our cells are never quiet. They are constantly talking, sending and receiving signals, making decisions. In fact, as you're sitting here right now, listening to this, countless conversations just like the one we talked about are happening inside of you. It kind of makes you wonder, doesn't it? What are they all saying right now?